interest of time, I'll get going. I can't tell how many we have, but it seems like we've got critical math. Great, so my name is Max Saito. I'm also one of the organizers, and welcome to the session on intercalibration. I'll be giving two brief talks, one on the introduction to intercalibration in biogeoscapes and another on the update for the ocean metaproteomics intercomparison effort, and then I'll pass it off to Paul Berube to talk about the nucleic acids effort as well. And let's see if I can get this going. There we go. So, you know, just to begin with the why intercalibrate question, you know, getting accurate values requires intercalibration efforts to using reference or consensus standards. That's sort of the general approach. And if we think about the definitions of from analytical chemistry, what accuracy and precision means, ideally we would like to be accurate and precise, but one can be accurate but not very precise with lower precision, or be accurate, not accurate but very precise with good reproducibility but not accuracy. And then also one could be not accurate and not precise. And to get into this upper left corner, of course, takes some effort and work. If one uses reference standards to intercalibrate, that enables a number of things. In particular, that allows us to use different analytical methods, and it allows different analysts to be involved, and allows new methods to be developed and incorporated. And we imagine an international program such as Biogeoscapes will have to have all three of these elements. And if we're interested in measurements across large ocean distances, as well as comparison of measurements across months to years, having this kind of accuracy is really important. And we can also consider the kinds of measurements, and this may be something that may be of interest to discuss a little bit in the breakout groups. A lot of times in omic measurements, relative measurements are used, whereas it's also possible to do absolute measurements where we calibrate and measure in copies per liter or moles per liter or reaction rate in moles per hour. And this could be really valuable when you're comparing against a changing biological matrix. And in doing so, it can enable stoichiometric analysis and rate estimates. And so I think sort of a take-home message that we'd love for people to take away from this is that at a continual intercalibration and standards effort and an internationally interoperable data system, which is something we're not really touching on that much in this session. But both of those things are perhaps really the foundation for an international biogeoscapes big data type program. You know, one can imagine we would have the ability to produce these kinds of 3D maps that GeoTrace is producing. This is one for iron. Wouldn't it be amazing to imagine this kind of distribution for key organisms in the ocean, SEP05, Baumarchiata, SAR11, Prochlorococcus diatoms, dinoflagellates, as well as biogeochemical rates or enzymes and metabolites and biomarkers and nutrient stress. I'm just throwing out some possibilities that's something that Biogeoscapes could do. And I think in order to realize this kind of global goal where multiple cruises are combined, we really need a solid intercalibration and data effort. All that said, intercalibration is difficult and a bit scary, but it really does have many benefits. And one thing that we saw in GeoTraces that is sort of a surprise benefit is that it really builds community. And you know, I think the trace metal community prior to intercalibration and GeoTraces was a really competitive community, and the intercalibration developed a level of trust and collegiality that was sort of an unexpected benefit. As Allison talked about yesterday, you know, this began with the SAFE program. This is an example of hundreds of measurements, I'm sorry, hundreds of bottles and the dissolved iron being measured them on the original SAFE standards. This was a program led by Ken Johnson in the early 2000s. Within GeoTraces, there's a standards intercalibration committee, and that is a standing committee that basically stands between data submission and the intermediate data product. That committee reviews every data set's intercalibration report before it can be accepted into the IDP. So that's really where the quality control comes in, and making sure that data that's coming into the program is you know, sufficiently rigorous in order to be included. And here's from the GeoTraces website what some of this looks like. These are various intercalibration samples that were produced and distributed. There are a number of elements, and there are reports about what the intercalibration consensus value is. And this is a sort of a interesting and key point. These are often consensus values because they're not necessarily certified standards, in many cases because we don't actually know what the exact concentration is ahead of time. But instead, the community gets together and all measures the sample, as shown by this list of analysts here. It continues on to another page I cropped off. But that results in what we call these consensus standards that then can be used to be a comparison for new data that's coming into the program. And then these are, updated as of this point in time, and new standards have to be created and periodically distributed. There have been a lot of publications that have come out of intercalibration efforts, so it is an academic effort in and of itself. This is just a few of the ones I grabbed. My apologies to others that I have left out, but there's some key ones here, in particular from people who participate in the meeting, like Kristen Buck and colleagues. And there's a paper on the sampling systems themselves. The particulate metal group, I think, did a really amazing job in geotraces. There weren't that many particulate measurements prior to geotraces, and their intercalibration efforts really lifted that to being a really valuable data type. And also looking at different, filter types and things like that. They did a lot of hard work there. I also want to do a shout out for this nitrogen fixation critical review that came out. So other communities beyond geotraces have been organizing as well. So to conclude this first part of the talk, there's really important work that needs to be done. And in the spirit of it being Veterans Day, I found this I Want You poster that came from World War I, a recruitment poster. And so our message from this meeting is that we encourage people to participate in intercalibration efforts and workshops. Many mines and labs really do make a difference. If there's some element or parameter or system that you don't see some intercalibration effort for, but you think could be really valuable in biogeoscapes, we encourage you to organize one. And I think we need to also recognize that there's some patience needed here. It will take multiple efforts perhaps to create consensus around parameters and values, but it will be worth it. It will create community confidence and trust. And I was cleaning my office last year and I found this note from Ken Brulin from when I was a graduate student that he put on my desk at a meeting saying, Mac, we need your cobalt data for both SAFE and GeoTraces standards. I would really like to arrive at a consensus value for cobalt. And it reminded me of this poster as well. And he was one of the heroes who helped lead the intercalibration of the bioactive metals. So with that, I'm going to transition to a update on the Ocean Metaproteomics Intercomparison meeting results. And this is a meeting that was just held a couple months ago, and a project we started in 2018 with OCB support. The project team is Matt McKilvin and Jackie Saunders and myself, and we have a great advisory committee from Dasha Leary from the Naval Research Lab, Mike Yannick from College of Charleston, and Ben Neely from NIST, who I believe is participating in the meeting as well today. And thanks to the program office. And one thing we wanted to really point out is that this is a volunteer effort. OCB supported our sample shipment and the workshop, but basically the work people have put in and the analyses was all volunteer or piggybacked on other efforts. Also, we managed to recruit a number of participants from outside ocean sciences, and that really helped, I think, improve the quality of the science that we were doing. So just to lay the stage, metaproteomics is a valuable newish data type of the omics data types. And I just went and grabbed a bunch of titles of papers, in particular some of the participants of the meeting here, to highlight that there's active work and real science being done with metaproteomics. Proteins are valuable in that they add information about function, for example, nutrient transport, reaction catalysis, and internal and external sensing, as well as regulation, to name some of the functions that proteins are important in. And they can capture adaptive, responsive to to environmental stresses, and those can actually correlate with transcripts. And we have a modeling study that we just put out. It's on bioRxiv and will come out in Journal of Proteome Research that visits this age-old question of do transcripts and proteins correlate. Also, proteins have the potential to get to be calibrated for absolute quantities and then be used to estimate potential reaction rates. And this is an example of a study that we did last year as well, trying to estimate the nitrate oxidation reaction rate using the enzyme measurements directly and using kinetic parameters. So with that said, our meeting this fall and the project had some overarching goals, and That was to provide community confidence in the measurements, and not just to ourselves, but because this is a relatively new kind of data type, we wanted to provide confidence to the ocean science community. In addition, we wanted to continue our community building and knowledge transfer efforts. This effort was piggybacked on or followed on the effort of a data sharing workshop we held in 2018 that was part of an EarthCube project. And here's a picture from that effort and a diagram we made from a review paper and a paper that went into the ocean best practices. And one thing we realized that is many of the researchers who were getting together on this did not know each other very well or hadn't met at all yet. And so it really was a community building effort for us. And from that, we were able to launch this intercomparison efforts. And these are the labs that participated in the intercomparison effort shown here, and people participating in the meeting as well that we had this fall. So lots of people, primarily focused on the US, but we did bring in a few international participants as well. And so the process involved sample collection of biomass from the Bats time series station using McLean pumps. And we had 2 McLean pumps that we chained together and had 4 filter heads. And then we took those filters and sliced them into eighths. Each filter head got about 200 liters on it. So each slice had about 20 to 30 liters on it. I have to show pictures of the ship because when I show this to chemical communities and other communities, the first question they ask is always, did you go on a ship? And which we love, you know, but it just to show the cultural differences that we are trying to span. And so this is our experimental design. We had global metaproteomics as our primary analyte of interest, and we did a technique called data dependent acquisition, or DDA. And we asked everyone to produce triplicate analyses using a one-dimensional chromatography that would allow us to produce comparable results. We also had, we allowed other approaches, but we made them optional. Some labs like to do 2D or gas phase fractionation. We also proposed a targeted metaproteomics, but we actually didn't have submissions for that at this time. And so that's something we're going to try and follow up on. And we also had a third informatics analysis component. And so those, the first and third, the process is shown in this flow chart. And what we found is that the raw data submitted for the first one was particularly useful because then we could put it through a single reanalysis pipeline. And I'll show those results today. I won't show any of the other results in the interest of time. We had a blinded study. The files were anonymized, and the labs were given ID numbers. We had a custom metagenomic database that came from one of the sample splits that was generously done by the Naval Research Laboratory and annotated by Jackie Saunders. Yeah, I mentioned these last 2 points already. So here are some of the results. We had 9 labs submitting data. A couple labs had some interferences and extraction problems, but we got data out of most of the laboratories and a large number of peptides and proteins. We were able to get raw data from 6 of the laboratories that allowed us to do our single informatic pipeline comparison. And that gave us almost 36,000 unique peptides and between 4 and 10,000 peptides shared in pairwise comparisons. And here are the pairwise comparisons between the 6 laboratories shown here. One thing that we realized in this analysis is that there's a very long tail of detection because of this DDA approach that we use, the data dependent approach. And that's because these samples are incredibly complex and there's many low abundance proteins. And when we limited everybody to doing a one-dimensional analysis so we could have reproducibility and comparability of methods, that also meant that there was sort of a stochasticity in the long tail of what was detected. And so there's more variability there. And that's really as a result of our experimental design more than anything else, we think. But when we look at the resulting protein identifications and the spectral counts attributed to them, we can see really good lab-to-lab comparisons. Here's 6 labs against each other. The ones in the diagonal here are lab against themselves, so perfect match. But you can see in general, just visually, we have a nice relationship between all the proteins there. And then we did 2-way linear regressions across all of these pairwise comparisons as well, and we're getting R-squared values from 0.4 to 0.7. So that's pretty encouraging, we felt. When we looked at the taxonomic assignment of those proteins using our single database as well, it looked really good. We see that within each lab, we get very nice precision. And then between labs, we're getting some nice consensus in the major taxonomic groups that are showing up. So that was very encouraging to all of us as well. And we also did a Bray-Curtis analysis of the reprocessed data just for the most abundant proteins, the top 1,000 proteins. And this showed 60 to 80% similarity between most lab groups, as shown here in these pairwise comparisons as well. And this is analysis done by Jackie Saunders. So we were really encouraged by this, and one of the efforts is, can we start combining our data globally for sharing and discovery? And we have our community's built the Ocean Protein Portal that has data from 7 different lab groups that have submitted that allows people, non-protein experts, to come in and interrogate data for questions of interest and visualize it, as well as pull sequences out and things like that. And so this intercalibration, I think, will really greatly help this data sharing effort. And then finally, looking forward, there's a lot to do. We're just getting going, I think, in terms of this. First of all, we have to write the report and write a peer-reviewed manuscript. We plan to deposit the raw files to allow future intercomparisons, so people can take their data and run it through their pipelines and see if they get the same results with our raw data files if we want to compare their pipelines. There's a lot to do for future intercalibration efforts. We called this an intercomparison because it was, it wasn't really an intercalibration yet, but clearly trying to get the targeted metaproteomics intercalibrated would be amazing using stable isotopically labeled peptides that we distribute. There's a lot of interest in a new technique called data independent acquisition, and starting to intercalibrate that would be really valuable. There's a lot more work to do on informatic intercomparisons. And there's more engagement we can do with international participants. In particular, we've recently interacted with the International Metaproteomics Initiative. They're primarily more biomedical, human gut microbiome and other communities, but they're really interested in partnering with the oceans community and excited about that. So I think that's a really great partnership. And finally, we need more intercomparison samples. And one possibility is to leverage the new CLIO AUV, which is really capable of collecting a ton of samples simultaneously. So with that, I'll wrap this up and just acknowledge again the workshop participants, most importantly, and thank you to those who are attending the meeting today from that effort, as well as OCB and NASA and NSF for their support, our organizing team and advisory committee. And I think instead of taking questions now,
I was going to say thanks, Mac, and pass it over to Paul. And please put your questions in the chat for Mac. We can address them in a short question and answer after Paul's talk.
Okay, can everyone hear me?
Yep.
All right, good. Thanks, Mac, for that really great presentation, and it's a good setup in terms of why intercalibration and standardization is super important for launching something of the scale of biogeoscapes. And in terms of trying to tackle the problem in terms of nucleic acids, this effort really came out of that 2018 meeting where Biogeoscapes was formulated and some national meetings had started. But we realized at the time that if we're going to get something like this off the ground, we do need to have a roadmap towards dealing with intercalibration and standards for ocean nucleic acid omics. So Adrian mentioned this yesterday, but in 2020, we had a meeting at UNC, just basically to get the ball rolling. And we brought in experts from around the country, including a representative from NIST, Steve Jackson, and really to solicit their ideas in terms of what the main issues are, the roadblocks in terms of moving this forward. I also want to thank the rest of the organizing committee down at the bottom. And Heather, I don't know if you want to just plop the link to the omics workshop in chat, just in case people want to look at who is there and look at, all the presentations are uploaded to that OCB website. Let's see. So, I mean, really the idea is that we want to, if we're going to intercompare and data omics data, nucleic acids from multiple countries, multiple labs, multiple workflows. We want to know that people are getting the same answer from the same reference material. And this will long-term enable us to do better analyses. It'll enable reuse and facilitate the long-term impact of these data types. Specifically for the workshop, we're talking about amplicons, metagenomics, and metatranscriptomics data. And some of the key tasks we focused on were to identify any sources of variability and bias that might come out of any of these omics production pipelines, to prioritize where we want to put our efforts in terms of trying to assess these variability and biases in the pipelines. And then moving forward, plan our research activities to actually get empirical data on what those biases are, and finally try to bring in the broader community in terms of a intercalibration effort to actually see how well different laboratories pipelines compare, to again build that trust and get the community involved in this effort. So in terms of what the workshop participants viewed as the key points of variability within these amplicon sequencing and metagenomics and metatranscriptomics pipelines, there's a fairly broad consensus in terms of the upstream parts of the pathway perhaps having the greatest impact. Again, these are perceptions based on the experts in the room's experience with these methods. Now, again, so what we're dealing with here is a very complex microbial community. We're dealing with bacteria, archaea, eukaryotes, different sizes, different susceptibility to lysis or capture on filters. So it was thought that perhaps some of the greater sources of variability might be in what we do in the field. So sample collection, whether you're getting it from a Niskin bottle or a pump, whether or not shear forces might cause loss of some of the biological material during sample collection. Using some of these newer AUVs like Clio, and enabling collection of large enough volumes so you can get detection of rare taxa. In terms of sample processing, whether you're going to use vacuum filtration or peristaltic pumps or pressurized filtration, the types of media you put the samples on, pore sizes, filter types, with material with filters, whether it's a Stereovex capsule or a disc filter. So, and then further on, sample storage. So, I mean, there's a lot of stuff going on right at the beginning of any pipeline that I think people agree that we need to pay a lot of attention to. When we get to DNA extraction, obviously there's different susceptibilities of cells to lysis methods, whether it's chemical or enzymatic or mechanical. So there's definitely things we need to do in terms of looking at variability and bias associated with how these nucleic acids are extracted from any biological material on these filters. And we get down to like sequencing and bioinformatics, things perhaps look a little better. Certainly there's biases known to occur with different sequencing platforms or different library preparation methods. But we can deal with that. We can box it off in terms of, if we start a intercalibrate or a trying to develop best practices and identify variables that are influencing the data, final data products. We can start with just looking at a single sequencing center or a single library preparation method, and then later on branch out and compare sequencing centers, for instance. Bioinformatics, I love this quote from Harriet, that it's certainly essential for intercalibration, but not likely our bottleneck. That's not to say that there are challenges with the informatics, certainly there are, but it's tractable. Moving on, we did also talk about the need for standards that enable quantification, QC and QA to further help with methods development. And there's several types of materials we could use for these standards. These include mock communities that are cell-based. So you can think of a collection of cultured representative marine microbes that can be used for protocol validation and methods development. There could also be a collection of cultured non-marine cells that could actually be added to samples to help with quantification. Certainly there's a lot of work to be done along those lines, and there's active ongoing efforts. And another alternative that could be perhaps more easily deployed are using purified nucleic acids in a similar fashion. So either something that's not found in the marine environment, or trying to develop perhaps clone libraries that are representative of the types of sequences we would find in marine systems. And these can be used as external, internal standards. So external standards would be key for assessing run-to-run variation in terms of sequencing. And I would foresee that in any BioGeoScapes program that it might be a requirement for every sequencing run to have a mock community on it, just to assess any differences between different sequencing runs. And then finally, with the internal standards, it's a key to getting quantitative data out of this. It's going to vary depending on whether you're looking at amplicon sequencing or metagenomics or metatranscriptomics. But for instance, and this has been done before for bacterial metagenomics and metatranscriptomics, but you can look at genes per mil seawater or genome equivalents per mil seawater. You can start normalizing transcripts to gene copy numbers per mil of seawater. And that's going to be really powerful, I think, moving on to the future. Now, in terms of actually moving into the phase of trying to get empirical data on this, the workshop, and there's ongoing discussion in terms of actually building out this effort, but one possible option is to break this up into having first a small group of dedicated laboratories that want to spearhead this and essentially do a thorough assessment of all these different points in these pipelines that could contribute to variability between different laboratories, and also just bias in terms of trying to get to that true value of what the microbial community looks like. So an example of this would be to set up a small program where you would get homogenized seawater from different environments, use different methodologies for collection of the material. Again, focusing on these upstream parts of the pathway that the workshop participants thought might be most important to understanding what's happening here. And you can see that it can explode out pretty readily with the different levels and the hierarchy of what's happening in these pipelines. But we do think it's tractable. And if we focus on what we think might be the key aspects of the pipelines. And then that could help us start developing these best practices, and then eventually move on to doing an actual intercalibration with the community, bring them in. Here we could provide reference material to labs as well as mock communities, and actually get an assessment of what precision and accuracy looks like between different labs. And this could be done just US or hopefully pulling in international collaborators, as well as ongoing efforts, perhaps with like Atlantico that was mentioned yesterday. And these people could use their own laboratory methods and sequencing platforms and just putting them on their own sequencing runs, which would also enable us, and this is what our representative from NIST talked about, in terms of having fine-grained information about the methods they're using to find out what are other key features of either extraction or sequencing that are contributing to lab variability. And then ultimately, this information can help us guide what we think are acceptable levels of differences between laboratories in terms of what we think would be compliance with any intercalibration for nucleic acid omics. So ultimately, I think we still have a long way to go, and it's going to take a huge community effort, but I think we're in a good spot. And hopefully, just taking this on will move the broader field. I mean, not just for biogeoscapes, but for other microbiome sciences. So if you have questions or ideas or input, please come talk to us. So there's a link right there for getting in touch with the organizers, as well as any of the attendees at the workshop. So, thank you.
Great. Thank you, Paul. Thank you, Mac, for some excellent talks here. So we are running a little short on time because we do want to make sure we have enough time for the breakout groups. But we do have time for perhaps one question. And then we encourage you guys to please just go ahead and keep including questions for Mac and Paul in the chat. And we'll have some time at the end of today's breakout session and example talks to come back to these. But maybe just looking at the chat here, there is a question for Mac from Bob Morris. Bob, do you want to just ask your question?
Sorry, yeah, hi. Hi, Mac. I was just curious in your own work if you've ever tried adding organisms at the beginning or putting purified proteins in, sort of like RNA standards, and what kind of, if you've done that, how it worked out.
Yeah, that's a great question. We haven't done it at the beginning, but we do regularly use stabilized labeled peptides that are digested that we add at the end to calibrate. But I think in the protein quant world, some people have explored adding whole proteins at the beginning and then following them through. Whole organisms is an interesting concept because it would add sample complexity, but the sample's already pretty complex already. So, yeah. Thank you.
Thank you.
Was someone? Okay. We do want to get everyone into breakout rooms to have more people have a chance to talk, and we'll circle back to some of the questions that came up during the intercalibration talks at the end of the day. So this is your charge for the next half hour. You're going to be split into groups, and we're going to use Jamboards again. And these are the 3 questions we'd like you to consider. What hypotheses could be addressed by Biogeoscapes program towards the goal of coalescing around a high-level motivation? What are compelling questions that can't be addressed by a single group and really require a coordinated effort? And following on from these talks, what are the key calibration needs to help support and realize those science goals? So try and maximize input in your breakout discussions. Jamboard links are going to be provided in the chat. We'll have a couple breakout rooms per Jamboard to hopefully avoid the problem that we had yesterday with a Jamboard jam. So a reminder of what we want you to do with these Jamboards. Use the Jamboard assigned for your breakout room number. Add your participant names at the top. Everyone, or you can assign a note taker, should capture the discussion. And please create a summary sticky to try and summarize your key points. This will help the organizers out a lot. And also feel free to email the organizers if there's anything you think wasn't captured or you didn't have enough room in the Jamboard. Your homework is to look at the Jamboards at the end of the day. We're going to use these to help write the report. And just a reminder, Mac introduced this idea yesterday to consider starting your breakout room with the circle process. So have each member of the group give their initial thoughts on the questions to try and help promote discussion and equal participation. Allow everyone an opportunity to speak. Don't interrupt. Speak with intention. Don't just ramble. Be respectful. Listen with attention. And tend to the well-being of the group. Okay, and just a quick summary of the remains of the day for us. We're going to have breakout sessions for about a half hour. We did have a 10-minute break planned. We're going to try and cut that short by about 5 minutes, so give you an extra 5 minutes in these discussion rooms. Take a 5-minute break, and then we'll come back to see some great science talks at 3:10 Eastern Time. All right, so you're going to be assigned to breakout rooms, and make sure to grab the Jamboard link before you head off into your breakout room. I'll linger here just another second in case there's any questions.
Hi, Heather and May. I was in the same room as Naomi.
Yeah, I was in the same room as Ben.
Okay.
That was I was in room 1, so I guess
Ben is in 2.
Okay.
2.
Do we have anyone 3? I'm going to move
Adrian.
one of you to 3.
Okay.
And Adrian, let's put you in.
Eight and 9
5.
have a low number of people.
Let's put Adrian in 5. There's only 7 people in 5.
Okay.
Oh, wait, look at
Yeah, 8 and
9.
9
9
are
is
low.
really low. People must be dropping
Or
off.
9 and 10.
All right, I'll move you to one of them.
Yeah, you can combine rooms if you think people are dropping off.
There we go. Kim, do you want to be moved somewhere? Oh, gone. It's nice that you can access the chat from the breakout rooms. I didn't think you could do that. I thought you could
think-
only,
They change stuff a lot.
yeah.
Remember early in the WebEx days, like every 2 weeks, we couldn't get something to work because it was different. I think they're still maybe slower, but I noticed.
Okay, so I think we can go ahead and get started here. So hopefully you had productive breakout sessions. And again, if you can just provide a summary of your breakout group discussion, that would be excellent in the Jam boards, and we'll come back to that and have a summary of those perhaps at the start of tomorrow's workshop day. So what we're planning to do now is just have a series of research talks that we basically identified the speakers from the registrants of the workshop. And essentially what we're hoping to do is provide some examples of what at least the organizing committee felt would be excellent sort of approaches towards looking at biogeoscapes like science that perhaps would come out of the program. And so our first speaker today is Dr. Scott Gifford, who's an associate professor, colleague with mine actually at University of North Carolina in Chapel Hill in the Earth and Marine and Environmental Sciences department. And Scott's going to talk to us about integrating quantitative genomics and nutrient metabolism, HNLC Synechococcus and the nitrogen and iron cycles.
Okay, great. Thanks, Adrian. Yeah, and so before I start, let me just give a quick shout out to my 2 graduate students here, Garrett Sharp and Liang Zhao Pitcher, who did a lot of the work that I'm going to talk about today. And so in particular, I'm going to focus on this using quantitative genomics to really try to understand nutrient cycles, and using our work in the exports program as an example of that and how it fits in with biogeoscapes. And so if you're unfamiliar with exports, this is a NASA-funded program to try to really understand what's going on in the surface ocean, in particularly to understand net primary production and how its fate. You know, where is it being exported, and how is it impacting the ocean carbon cycle? And so as part of the program, we've got 2 field campaigns. We just had one in the North Atlantic, and we also had this one in 2018 in the Station P, and that's what I'm going to focus on today. So this is a multi-investigator program. There's 40 plus investigators involved, collecting a wide variety of measurements. And when we were out in Station P, we had 2 research vessels occupying one site. So in biogeoscapes, I think we're talking a lot about spatial diversity. Here, we've got a big team just sitting in a Lagrangian study, basically following this Lagrangian float, trying to characterize a given water mass. So this actually comes out of the breakout discussions that we had yesterday. one of the things that it's really exciting about being part of this project is that we have all these investigators collecting a wide variety of measurements. And again, when we were in the breakout sessions yesterday, it came up that in BioGeoscapes looking at omics, chemical measurements, and really the metabolism linking those two. And exports has been a really kind of interesting experience to see how these metabolic measurements that are kind of classical we made for a while, their variability and how their different time scales affect this kind of linkage between the omics. And so I'm putting this slide up here. This is work by Heather McNair, Tatiana Rush, and others in the exports crew to bring all of our exports data together to try to form this metabolism model. And I think we can leverage that a lot for the BioGeoscapes program. So in particular, our group was looking at the microbial community, and we were on the cruise 2018 at Station P following this Lagrangian float. And basically over this 30-day cruise, we sampled for metagenomics and metatranscriptomics, really kind of up in the surface water column. So we've got by now what's a pretty standard type of metagenomics protocol, collect the samples, flash read in nitrogen. But right before we do that extraction, we add a series of these internal standards that Paul was just talking about. They go through the whole omics process. And then once we get to the analysis, if we do something like an assembly independent process, we can annotate those reads, but we can use the internal standards then to take those reads and convert them to some volumetric abundance, taxa per liter or genes per liter. We can also do an assembly dependent process where we're taking that data, assembling genomes, MAGs, from the data. But again, because we have these internal standards, we can take that MAG data and start to put it on an absolute abundance scale and really try to get at things like what are the MAGs representing in terms of populations per liter. And I'm going to focus on that today. So, real quick here, if you look at Station P, looking at just kind of metagenomic taxa assignments to the reads, we get a really kind of typical what you'd expect with a surface community going down through the water column, LASR11, flavos, gammas, rosavectors, the usual suspects. Then as you move down deeper in the water column, of course, things like Archaea, Biome, Globus, et cetera. So what we really kind of zoomed in on here is that at Station P, there's a decent Synechococcus population that exists kind of deep in the water column. And so, or I should say, a little bit deep in the water column. If we start to bring in the hydrography data from one of these profiles, what you'll see is that black line representing temperature, we got the mix layer, and then just below the mix layer, if this is our fluorescence, we got a DCM. And here in these green dots, I'm showing flow cytometry data from Jason Graff, looking at Synechococcus populations. And so the reason we started to focus in on this is because, I mentioned we're trying to do this quantitative data. And one of the great things about, having flow cytometry is to get these absolute Synechococcus abundances. And so we took from our metagenomes, we took a single copy REC-A gene for Synechococcus, converted it to these absolute abundances, that's shown here on this axis, and then compared it to Jason's Synechococcus flow cytometry data. And what you'll see is over the cruise, we have a really nice fit between the flow cytometry and the meta-derived abundances. This dashed line is the one-to-one line, and then the red line is our fit. So really giving us confidence that the accuracy of this metagenomic abundance is informative. We can then go down and look through the water column at these different Synechococcus groups. And so when we looked at the REC-A, we saw that there really kind of were two main groups, a clade 1 and a clade 4. And again, now we're showing the metagenome derived abundances down through the water column. And what we can see is these major clade 1 and 4, which we expect at higher latitudes, really dominate the population. Okay, so that was kind of just looking at single copy genes within the metagenomes, but of course then we can go and assemble the genomes. And so we took a hybrid assembly approach where we did short Illumina reads and long nanopore reads. And from that, we were able to assemble two genomes of Synechococcus. And those two genomes here, SYN-SB1, SYN-SB2, they represented each of those dominant clades that we found out there, clade 4 and clade 1. High completeness, 96% complete, low contamination, less than 1%. So because we have those internal standards there, again, we're trying to work towards that quantitative ability. We can go back to this same depth profile that I showed before, but now what I'm putting on there is the MAG volumetric abundances, right? And so what we see is that our MAGs that we recover when we look at their volumetric abundances, they're really right on with what we're seeing in the REC-A abundances for those clades. And allows us to say, that the MAGs are really capturing the dominant Synechococcus populations that are out there. And that's important because now when we look at the genome content, it gives us confidence that we're actually saying something about the major microbial populations of Synechococcus that are in the water column. So, and so when we did that, we said we started out, we know we're in an HLC region out there, low iron, look for an iron signal, and we didn't really see one. We didn't see anything specific to station P, I should say, for these Synechococcus MAGs. What we did see is an interesting signal in nitrogen metabolism. And so cyanobacteria have a pretty conserved locus where they've got a lot of their nitrogen genes, things like urease here in green, nitrate transport and reductase in red, or nitrite transport and reductase in blue. And what was interesting about our MAGs, which I'm showing here in this blue line, have urease, one copy, but they were missing both their nitrate transport and reductase, as well as their nitrite transport and reductase. And of course, this is pretty surprising because they're in an HNLC region where there's a ton of nitrate and nitrate in the system. We expanded that analysis then to look at other high quality, nearly complete Synechococcus genomes. And at this point, it turns out that these are the only known of those genomes to be missing both the nitrate and nitrate genes for taking this up. We've done a lot of work to say, because this is a MAG, is this like an assembly issue that we're missing things? And don't have time to get into it, but yes, we saw that, including because we have those long read data, we have that nitrogen region and they're also missing those genes. So we feel confident about that. But we said, okay, just set that aside, the assembly, let's look at the unassembled metagenome data. And we can look at either enrichment or depletion of these nitrate genes in the unassembled metagenomes. And so we did that. Garrett went and he looked at things like nitrate reductase abundances in relation to something like Synechococcus rec A. And so what you'll see coming out of that is if you look at the black for nitrate reductase, it's typically found in the unassembled metagenomes at station P at about 1 for every 10 Synechococcus rec A copy. And so fitting with our observations with the MAG, the nitrate reductase is really depleted in the system. You compare that with something like these gray ones here, that's that urease genes that we see in the MAGs at 1 copy. And you also see in the unassembled genome that same thing. What I didn't mention was that the Synechococcus MAGs tend to be enriched ammonia transporters. They have 2 ammonia transporters in the genomes. And again, when we looked at the unassembled data, we can see these red dots here are the ammonia transporters present at 2 copies per cell. So, it's nice to have this genomics data, but what really I think is exciting and fun for us as a group is that we can take this genomics data and then work with all the other exports crew to try to put this in context of chemical rates and stocks, and which is really what we're trying to go for with the BioGeoscapes program. And so what I'm showing on here on the right is just a sample of some of the measurements that we've taken as part of exports, things like chlorophyll biomass, carbon uptake, nitrate. This is from Adrian's group. We've got that flow cytometry from Jason. We've got things like ammonia uptake, ammonia concentrations from Allison. And that really allows us to kind of try to fill out this story. And, just briefly, Station P, we know iron is low. Paul has really kind of been a leader in looking at why do cyanobacteria contain these different nitrate genes. Part of that is definitely that there's an iron requirement. And at Station P, what we're seeing with this other data is there's actually a lot of ammonia. There's not much competition with things like Prochlorococcus, which just isn't up there, probably reducing the pressure that you need to maintain these nitrate and nitrate genes in this particular system, allowing these Synechococcus to just lose it. I just want to end with this here, because what Garrett did is he took this kind of idea of looking at copies for these nitrogen genes and applied it to the Terra Oceans data set. And so we looked at things like nitrate reductase or ammonia transporters, comparing exports versus Tara. And I think the most informative thing to look at is this graph over here, where again, you're looking at nitrate reductase copies per Synechococcus rec A's, right? And what you'll see is that those depleted gene copies of nitrate reductase correspond with different samples that come from these, enriched in nitrogen compared to iron type of samples. And here's our station P up here. And this, again, Kristen Buck provided us this data. She was on exports, and so we could leverage that directly. But then we could look at this Terra data set to kind of see that these other red dots here are from other HNLC regions, allowing us to send this observation that, you know, maybe what we're seeing in these HNLC regions is a depletion of these genes in Synechococcus. Okay, so just to kind of wrap up, what are some potential takeaways for the Biogeoscapes program? Well, so it's really critical that we have these omics, metabolic, and chemical data measured simultaneously. So, you know, when we look at this map here, this is all modeled data. And Adrian showed this before, where we have the Terra data sets and something like geotraces, and there's this big spatial separation. Whereas in exports, this was all put together, and that really helped us tell the story that we can. The other thing then is this quantitative data. You know, and Mac was mentioning this, Paul mentioned it about the SNI community, is that when we take and we put our omics measurements on a volumetric abundance, we can then start to put things at the same level that we're taking our stock and rate measurements. And that at least puts us at a level playing field to try to move forward with putting our data into models. And I think with that, I'll end there.
Great, thank you so much, Scott. Please put questions for Scott in the chat, and we're going to move straight into our next talk, which is Bethany Edwards from the University of California, Berkeley.
Hi, everyone. So today I'm going to be talking about integrating lipidomics with other analyses to capture some complex ocean biogeochemistry. So lipidomics is arguably the oldest omics subfield, and it's having a revolution due to advances in mass spectrometry. And this is also true for metabolomics and proteomics as well. You may be familiar with this graph looking at the characterization of different biomolecules, in particular organic carbon, which was put forth by Stu Wickham, Cindy Lee, John Hedges, with work from Stu Wickham's graduate students and postdocs at the turn of the century. And so we've been making these bulk measurements of amino acids, carbohydrates, lipids for a very long time, and looking at how they degrade with depth and time in the water column. If we look just at the lipids through a series of different types of extractions, derivatization steps, and then measuring these lipids with different types of mass specs and flame ion detectors, we were able to characterize the total fatty acid content of organic material, total sterols, wax esters, tags, in both the suspended particulate organic carbon and in sinking organic carbon. And so using kind of classic lipid chemistry in the 1980s, 1990s, and early 2000s. What modern lipidomics gives us is this push towards measuring all of these lipid subclasses in their intact forms. So not just looking at total fatty acids, but looking at fatty acids attached to a head group or attached to a triglyceride. And doing this in one, maybe two analytical runs without derivatization. So here we're looking at the lipidome of the diatom Phaeodactylum tricornutum, and you can see that we can analyze signaling lipids, structural lipids that make up the membrane bilayers, energy harvesting lipids like quinones and pigments, and then energy storage molecules like tags and wax esters. Because the lipidome and the metabolome are the culmination of the central dogma of molecular biology, we can get a lot of information about chemical reaction rates if we measure these pools over time. We can also say a lot about the biogeochemistry of the system by measuring the culmination of DNA being transcribed into RNA, which gets translated into proteins, and then those proteins facilitate chemical reactions. Lipids have long been used as biomarkers, and they're longer lived than nucleic acids. And so we can use things like, some examples are TEX86 and UK37, which are used to look at sea surface temperature in the geological past. In the modern ocean, some lipids have been found to be more sensitive than DNA biomarkers. The example I'm thinking of is work by Christian Laber in 2018, who was a student with Kay Beidel and Kim Thimatricon, looking at viral glycosphingolipids to study viral infection in EHUCs. And they found that these lipid biomarkers were more sensitive than the DNA biomarkers for indicating kind of this post-infection state of a bloom. And then lipids, when we compare lipids and metabolites to the transcripts that they're associated with, we see that there are diel offsets between gene expression and peak in metabolite and lipid concentrations. And a lot of work that was done by Scope out at Station Aloha has really highlighted these differences in this offset between transcription and these chemical reactions. So I would advocate that lipidomics and metabolomics should be included in the biogeoscapes model. We can think about the biological carbon pump. Here I'll take away the numbers so it's a little less chaotic up here, but we can use lipids, right, to look at lots of processes within the biological carbon pump. So things like structural lipids can tell us something about chemotaxonomy, biomarkers for viral infection, metabolism. Pigments have long been used. Chemtax has been used to say something about what does the phytoplankton community look like. Can also be used as biomarkers of bloom state by looking at the degradation products. And I'll be highlighting specifically how signaling molecules can be biomarkers of phytoplankton bloom dynamics and bloom states. I spent most of my career looking at chemical signaling via oxylipins, which are oxidized lipids. They're mostly produced by diatoms whenever the cells are stressed. And here's just a look at how these oxylipins produced by diatoms impact all these different players in the biological carbon pump. So we know from some field observations in the late 1990s that copepod grazing induces the production of these compounds. And then these compounds inhibit reproduction by copepods and can decrease copepod egg viability by about 80%. Matt Johnson and I did a series of experiments looking at microzooplankton grazing in culture and found that microzooplankton grazing also induced the production of oxylipins. And in turn, the oxylipins inhibited grazing and deterred microzooplankton grazing. And we also did these experiments with deck board incubations with natural communities and saw the same thing. We're finishing up a series of experiments with Kim Thimatricon at Rutgers, looking at viral infection of diatoms and how this induces the production of these oxylipin compounds. And we had done some deck board incubations, taking trap material, exposing them to these oxylipins. We saw this increase, stimulation of particle-associated bacteria, where respiration was increased, enzyme activity was also increased in response to these compounds, which is very different from the way that free-living bacteria respond to these compounds, both in culture and deck board incubations. We know from a series of culture experiments that eukaryotic phytoplankton that don't produce oxylipins are inhibited by these compounds. And so oxylipin signaling clearly has an effect on the biogeochemistry of the microbial food web. And it's not the only chemical signal out there, right? There's been work looking at DMS and how it impacts these ecosystems. Phytoplankton also make hormones, phytotoxins. So there's likely lots of other signals, chemical signals that are cascading through the food web. We'll look at a study off the coast of California where we've integrated across lipidomes, the particulate and dissolved lipidome with metatranscriptomic study to better understand how chemical signaling may be occurring in this system and how it might be linked to or is linked to diatom bloom decline and viral infection. Hannah Kranzler, who was a postdoc with Kim Thimatricon, she published a paper in 2019 that showed that this community that we were studying in 2013 in June, that the community at Point Reyes was regarded as kind of a post-infection state with regard to known diatom viruses. Whereas Monterey Bay, when we sampled it, was actively undergoing infection with regard to known diatom viruses. And there was a link to silica stress. We wanted to know, what can we learn by looking at the metaomes in an integrated way? Could we corroborate Hannah's story, and could we learn something new? So we looked at the 18S amplicon sequences of the eukaryotic community. We also looked at the cell-associated viral metatranscriptome. And so Hannah's paper was focused on these known diatom viruses. There was a lot of other transcripts mapping to, at Point Reyes, a lot of these unclassified single-stranded RNA viruses. We also looked at the particulate lipidome and the dissolved lipidome across these sites. To integrate across these data sets, we used data integration analysis for biomarker discovery using a latent variable approach, which allowed us to ask, what's the most discriminant feature in each of these metaomes? And what we found was that Point Reyes, which we considered post-infection with respect to diatom viruses, also had a strong link at that site. Here we're looking at kind of the relative expression of each of these components of these different omes at Monterey Bay and Peach, and Point Reyes in cyan, and offshore in gray. We found that these unclassified RNA viruses were linked to diatoms navicula and Thalassiocera. They're positively correlated with reads from these two species or genera, and also these oxidized triglycerides. We find that these compounds, oxidized triglycerides, are upregulated by diatoms whenever they're undergoing oxidative stress. At least we saw that in Phaeodactylum trichromutum. We also saw that there was the link to these allelopathic oxylipins. We happened to have some of these oxylipins with us when we were at sea. So we did some experiments where we added an analog to this compound here into some grazing experiments, and we saw a significant decrease in microzooplankton grazing. And we also added these compounds to incubations with trap material, and we saw a stimulation of particle-associated bacteria, their enzyme activity rates, and a slight decrease whenever we added these compounds to the free-living communities. While correlation isn't causation, it does give us some indications of particularly or potentially some host virus interactions that might be worthwhile looking into. We've also done some diel studies, sampling the lipidome every 12 hours for 5 days out here at the Mars station outside of Monterey Bay. And here we're looking at salinity and temperature over those 5 days, as well as the moon cycle. In night 4, there was a new moon, which would indicate higher tidal forcing. We saw that the particulate lipidome was significantly different between day and night at this site. In this one instance, where a night sample groups with all the day samples is the night of the new moon, and we have all this tidal forcing. And when we ask, which compounds are most significant for this clustering, this differentiation between night and day, we find the types of compounds that were more abundant during the day that Ben Van Mooy's group has seen diel signaling in the oligotrophic open ocean at Station Aloha. We also see a wax ester compound here that we think might be associated with diel vertical migrators. We're hoping to integrate this data with DNA data from Francisco Chavez's group, who was leading this cruise, and exploring more of these biomarkers for the systems. And then lastly, my lab group has been collaborating with Meredith White at Mook Sea Farm in Maine. And they've seen this emerging syndrome where larvae, oyster larvae, will ingest food, but they won't digest it. And we did some studies for them, and we found that there were many oxylipin compounds in their system. And depending on how they treated their incoming water with UV or non-UV, they could produce more of these oxylipins. We're hoping to get funding soon to continue doing this work and integrating genomics, so we can better understand, are there certain phytoplankton taxa, diatom taxa, that are associated with these oxylipin concentrations. And this is an emerging issue. It's not something that they had seen prior to 2020. Just to wrap up some future considerations as we talk about biogeoscapes and what it could be, is thinking about the time scale that we're sampling on. There's seasonal and diel variability, also the lifetime of blooms. If we're sampling at the beginning of a bloom versus as a bloom's declining, we might see very different things. How do we integrate across the entire biological carbon pump and microbial carbon pumps? Ideally, we would have people who are experts in virology, archaea, bacteria, phytoplankton, zooplankton. I've seen a number of papers now looking at marine fungi and how important they are, as well as looking at both sinking and suspended POC and characterizing the dissolved organic carbon pool as well with respect to metabolites. And then, as we've been talking about, these omics, rate measurements, geochemical characterization, we kind of need it all to tell a complete story. And then, is there an avenue to go back and do culture studies with isolates or these complex model systems that we can build? And lastly, the statistical techniques to integrate all of this information. And I'd just like to acknowledge all my co-authors and collaborators and funding agencies. And I will pass it off to the next person.
Thank you so much, Bethany. So again, just continue to place your questions in the chat. We're going to get to those after our last speaker. So our next speaker is Dr. Julie Granger, who's an associate professor at the University of Connecticut in the Department of Marine Sciences.
Thanks, Adrian. You can hear me? Have thumbs up. Okay. So thank you for this invitation, and very honored to present. Well, I would have been honored to present my work, but ultimately I'm going to present mostly Sarah Fawcett's work today, because I've just started to dabble in this method and I don't have a compelling story yet. And the idea here is to compel. So Sarah wanted to be here today, but it's way past her bedtime. So she texted me that, yeah, it's too late in Cape Town for this. So today I'm going to talk about how we can use flow cytometry coupled with nitrogen isotope techniques in order to glean information about who's using nitrate in the plankton community and who's relying on recycled sources. And this may give us some information about export production. And I think you'll see that it also gives a really interesting view of how nutrients are supplied to the euphotic zone, especially in how they're supplied to the stratified ocean. So this is a graph from Sarah Fawcett following John Casey and Mike Lomas. This is the Bermuda Atlantic Time Series. This is sort of the typical populations that you can sort by flow cytometry. So you have a large population of Prochlorococcus that you can identify, Senecacoccus, and then a group of pico eukaryotes, so very small eukaryotes, whose approximate size largely, a lot of it overlaps with Senecacoccus, and then, fewer nano eukaryotes. There's a lot of schmutz, part of which is probably heterotrophic bacteria and other sort of debris, which you could identify the heterotrophs, of course, with a stain. Now, during her doctorate with Bess Ward and Danny Sigmund, Sarah worked closely with Mike Lomas to develop a technique where using cell sorting flow cytometry in order to get at populations, these distinct populations, so collect these populations in distinct samples, and then she oxidized them to nitrate with persulfate, after which she fed them to denitrifying bacteria to convert nitrate to nitrous oxide gas, which becomes the analyte for nitrogen isotope ratios on an isotope ratio mass spectrometer. The beauty of this technique is that, for instance, in my lab, we can get down to 5 nanomoles of nitrogen. So with 4 liters of water at Bermuda filtered onto a 0.45 micron filter, you can sort enough cells to get triplicate analyses for all of these populations on an isotope ratio mass spectrometer. So this technique is about 300 to 1,000 times more sensitive than if you were just to collect populations on a filter, combust them, measure the N and N2 gas. So it's a very powerful way of looking at something that would probably be impossible otherwise unless you wanted to sort cells for the rest of your life. So to give you context, so if you're at the Bermuda Atlantic Time Series, of course, is a highly oligotrophic region. So nitrate and other nutrients are virtually absent from the surface ocean down to below the euphotic zone, which is about 100 meters. And then if you look at nitrate isotope ratios, these are profiles that Sarah measured. You can see that at the subsurface, the isotope composition of nitrate is between 2 and 3 per mil. And as you get into the base of the euphotic zone, you see partial nitrate assimilation. Now, remember here, we're in the hundreds of nanomolar range for nitrate. So Sarah's just showing off by even measuring these. I don't even bother below 1 micromolar. And for a long time, we assumed that this was due to autotrophic assimilation, but we're now realizing that here and in the North Pacific subtropical gyre, this is associated with AOU. So this looks to be heterotrophic nitrate assimilation at the subsurface, which if you followed any of Sarah's work, it's, she thinks it's TEP material. So carbon-rich gelatinous material that is getting digested by heterotrophs. But in any case, the important point here is that if there's an injection of nitrate into the surface by upwelling, you would expect it to be between 2 and 3 per mil. Now, if you measure the suspended particles at Bermuda, so remember here we're in the euphotic zone, there's 0 nitrate to be measured, or ammonium for that matter. If you collect, say a liter of water onto a glass fiber filter and measure what you collect by elemental analysis, you're about minus 0.2 per mil. Mark Altabet did this early on in the 80s, and we get the same answer now. Mark Altabet initially ascribed the difference between the suspended particles and the nitrate supply as reflecting recycled production. So this is largely a system that is sustained by recycled production, and there are fractionations to that from the excretion and bacterial degradation that cause the recycled N, so largely ammonium, to be isotopically deplete compared to the nitrate supply. Now, Sarah was, for the first time, able to look at the N15 specific to phytoplankton groups. And somewhat surprisingly, what we thought was entirely recycled production signal was it looked like the recycled production signal is even lower than what's in the suspended. So Perchlorococcus, which we presume does not assimilate nitrate, and that seems to bear out in all of these measurements that Sarah's done, sits at around minus 2 per mil in this profile. Senecacoccus does similar stuff to Prochlorococcus with small deviations. So this is summertime at Bermuda. And here we have the small eukaryotes, the pico-eukaryotes, who are not much larger than the Senecacoccus, yet seem to rely entirely on nitrate, at least in this profile. I still remember when these data came off the mass spectrometer. We were in the same lab, Sarah and I, when I was doing my postdoc, and I just could not believe it, because if you'd asked me how this was going to come out, I would never have guessed this, that the eukaryotes look to be assimilating only nitrate. And this was not a fluke. Sarah's done a gazillion of these measurements at Bermuda. So this was, I just showed you summer 2008. Here's summer 2009. there's a pattern that seems a little more sort of intuitive here, that at the surface, even the eukaryotes seem to be relying heavily on what we think is recycled nitrogen with a tiny bit of nitrate. But then when you get into the base of the euphotic zone, they're relying more and more on nitrate. But again, there's no nitrate to be measured at any of these depths, right? So it begs the question, how does nitrate get into the mix layer in the dead of summer? Don't have the answer to that, but it's there according to the cells, right? So another really sort of serendipitous and cool little thing that happens is so Mark Altabet in the 80s had shallow sediment traps deployed over an annual cycle at Bermuda. And the sinking flux of particulate nitrogen at Bermuda pretty much sits squarely at around 3.5, which is the N15 of the nitrate supply. So from there, so we're able to conclude that, well, what goes up comes back down in sediment traps. But then this also intimates, at least to a first order, that the eukaryotes that rely almost exclusively on nitrate in this profile are those that are dominantly sinking, or are the dominant drivers of biological pumping. So of course, I can hear you squirming, even though I can hear you. I can't hear you, but this is what these data suggest, right? So this is an example of work for one of Sarah's graduate students, Colicia Signania. And this is from the Agulhas current. So here, these data show something slightly different, which is a recent injection of nitrate into the mixed layer, where the phytoplankton have not yet had time to react or are partially reacting. So the Agulhas current is a very powerful current, right, that brings surface water from the Indian Ocean into the Atlantic. And usually the mixed layer from the subtropical gyre into the current follows the 25.5 isopycnol. But here there's some sort of a disturbance, according to Sarah, there seems to be a nitrate injection given that the mixed layer has sort of shoaled onto a higher surface. Now, if you look at nitrate per se, there's a little bit of nitrate that you can see in the profiles, but what's most interesting is if you turn to the isotopes, you can measure the N15 of the nitrate at the edge of the current and into the gyre. And you can see that there's a little bit of nitrate assimilation actually by the Senecacoccus here. And then there's been even more nitrate assimilation by the eukaryotes. But then a little further downstream in the current, where you can measure this recent nitrate injection, it doesn't look like the eukaryotes have yet had time to react, and because they were relying on recycled sources as well. So a bit of a different story there. So briefly, I showed you a method where you have sort of this naturally occurring short-lived tracer of new and recycled production. You can estimate an F ratio if you want based on the biomass of the relative groups and their N15. This tracer doesn't stimulate uptake. You could imagine getting a result by adding N15 nitrate in the oligotrophic ocean, where cells are primed to utilize nitrate, but there would have been no nitrate otherwise. You're looking specifically at phototroph, so you're not looking at heterotrophic material or detrital material, although you can stain the heterotrophs and get at their N15, so Sarah's worked that out. And this provides a way to record nitrate assimilation when nitrate is below detection. And I think more excitingly, perhaps, or equally excitingly, this provides a link between mesoscale and sub-mesoscale physics and biology, like the things you can't really measure in real time, but are occurring to biology. So you could expand this method, of course, to target specific populations. You can use polarized light to sort out coccolithophores. Apparently, you can stain silica to look at diatoms. I'm always trying to think of how to do mixotrophs. You can apparently stain vacuoles that are acidic and look for phototrophic mixotrophs. But also you can incorporate these kinds of things with other tracers, of course. A subsample can be divided to look at the metagenomic and metatranscriptome of a sample. You can get at N2P quotas fairly easily of certain populations. And given sort of deck board incubations, for instance, you could look at population-specific C14, phosphorus uptake, iron 55 uptake, you name it. Now, I tried to think of questions that I'm interested in, and perhaps other people will be interested in, that leverages the biogeoscapes idea, sort of I mean, from this work, the idea that which phytoplankton groups dominate new production is fairly pertinent. And do eukaryotes dominate export production? Of course, that's not, a lot of people would argue that they don't. And then I'm super interested in what's the impact of mixotrophy on the food web, and can we use these techniques jointly? So what I just presented today and what other people are offering through Biogeoscapes to figure out what is the role of mixotrophy in the food web in different realms of the ocean. More specifically to this method, there's always a difference between the recycled nitrogen that you see in Prochlorococcus and the nitrate, and that difference changes in different times, different regions, and it's a meaningful difference. There's something about it, but we haven't figured out what that is, and that's the kind of thing that one might want to tease out through both more measurements, of course, and modeling. And then the work I just presented goes directly to looking at what are the mechanisms of nutrient transport in the stratified mixed layer, which is still a big mystery. I'm interested in we see evidence of heterotrophic subsurface nitrate assimilation from when you have highly resolved anisotope profiles at the subsurface, which is related to this idea of negative preformed nutrients or the remineralization of glom, but that's something I'm very interested in, and that Biogeoscapes could come to bear on these types of questions. And also, is there such a thing as subsurface non-euphotic zone nitrogen fixation? And that's something too that I think some of the Biogeoscapes program with all the combined measurements could start to address. And with that, so I think this type of work leverages the strengths of Biogeoscapes because it's a means of linking community composition, gene expression, proteomes to function. It can involve multiple science groups. Sarah's a unicorn, she does it all. I'm just starting to dabble on the flow cytometer and I'm terrible at it so far, but this could involve mass spectrometry as well as all the omics, of course, would inform these types of questions and modeling. And the Biogeoscapes promises an increase in data density like geotraces. And I think these programs are vital to the research enterprise because sometimes you don't know what to look for until you find it. Hypothesis-driven science sometimes puts us in silos that and it's hard to propose not hypothesis-driven science to NSF, but the ability to just go and map the ocean for X, Y, and Z is a real privilege that leads to developing more hypotheses and more pointed questions about how the ocean works. And with that, I thank you for your attention.
Thank you, Julie. Thanks for ending with an ode. Our next speaker is Scott McCain. Scott's finishing up his PhD at Dalhousie, and he's going to be starting a postdoc at MIT very soon. Scott, are you ready to share your screen?
Can you see this?
I can see it. Let's see what happens when you put it in. All right, that looks great, and we can hear you. Take it away.
Okay. Thanks everybody thanks for the organizers for this opportunity. I'm really excited to be here. And I will admit this is the first talk I've ever given in sweatpants, so that's also quite exciting. So today I'm going to try and convince you of a way or a method that we've been trying to go from omics to rates. So omics are this really big data stream of mass spectrometry data and nucleic acid sequencing. Can sort of feel like a deluge sometimes. And one, I think, central challenge is how do we make this leap from going from omics to rates or some sort of time-dependent process? And that's really the ultimate goal of this talk is how we've been trying to make that leap. And in particular, I'm going to try and convince you that, or demonstrate one potential way that you can combine molecular and biochemical measurements and then maybe this could be done at a large scale, but we haven't really done that yet. But starting with a historical perspective, I think it's important to note that people have been really inferring rates from omics for decades. So here I'm showing a plot from the 1950s, looking at a proxy for ribosomes per cell and growth rate. And while the goal of this paper was not to really predict the growth rate given a certain amount of ribosomes, you can totally see that given these really nice correlations, they would have been able to do that. And from a different perspective, this paper that was published just a couple of years ago using machine learning to try and predict growth rate from gene expression was a really interesting approach. But again, this is a bit different than what we might be interested in, because this is typically from single organism cultures. And some really nice work from Mac and Mac Saito and others, many of whom are here today, used metaproteomics to try and constrain transformations of nitrogen in the Pacific Ocean. I think one general challenge with trying to go from omics to rates is that we don't always understand the mechanistic connections between gene expression and the different things that we care about. And I really say things because it could be as broad as you'd like to make it. You might be interested in nutrient quotas or nitrification rates or translation rates or growth rates, or really any process that you're interested in. But we don't always know how the production of a protein or a set of proteins actually underpins these sort of larger microbial behaviors or characteristics. And one approach that we've been doing is trying to build models to connect a bunch of processes together. And then that really helps us constrain the connection between gene expression and the things that personally I care about. And in particular, what we've been doing is connecting these models with metaproteomic data. And I think in general, coupling metaproteomic data with cellular modeling can be really powerful, and it can achieve two main goals. The first is that we can try and quantify different cellular costs and constraints. So for an example, we might be interested in, and others have been as well, what the membrane space available is for nutrient transporters. And so by coupling a model with metaproteomic data, we can try and get quantitative estimates for these sort of key cellular processes. And what I'm particularly excited about in this regard is that we can do this even with things that are super difficult to culture. So we can start to ask questions more about the cell biology of these different organisms by coupling a computational or mathematical model, and we wouldn't necessarily have to grow the organisms in the lab. But today I'm going to talk to you about a second goal, and not a second first goal, and that's how can we use this coupled approach to make inferences about rates and quotas. So in particular, I'm interested in iron and manganese, and I've been working on this proteomic allocation model for a Southern Ocean diatom. And if you're interested in the details of the model, we just published this paper in Science Advances just a couple of months ago. This is just a schematic for the model, but it's the main thing to take away from this is that there were several critical parameters that were estimated by leveraging metaproteome-derived diatom proteomes. So we're trying to use the metaproteomic data to try and estimate key unknowns and constrain this model. And then another thing to note, and it's more general, not necessarily for our model alone, but in general, a cellular model might have inputs of environmental characteristics, for example, dissolved iron and dissolved manganese concentrations. And it has outputs, in our case, of coarse-grained gene expression. So we were interested in sort of groups of proteins rather than individual proteins. And excitingly, it also has associated rates and quotas. So we can get in a gene expression profile and connect that to a growth rate or to an iron uptake rate. So the first thing you might be asking is this a realistic model? And does it produce realistic data? And we did this in many different ways in the paper. And if you have any questions about that, I'd love to chat with you. But in one specific way that we did this was compare it to cultured growth rates. So here what I'm showing you is growth rate data from Brooke Nunn's paper in +1, which is a diatom proteomics paper. And they looked at the difference in the proteomic response under low and high iron. And here I'm showing you the model in green, which is the posterior probability distribution. And then the actual growth rate in the culture is shown in blue. And this is the high iron and low iron treatment. And you can see that the model does a pretty good job, at least for the high and low iron treatments, in recovering or reproducing similar growth rates. But one thing that's really important here to note is that there's no model training on the growth rate data. So we didn't actually estimate any of these parameters on the growth rate data, we only estimated parameters using the proteomic data. And maybe that will give you some faith that this model it has some realistic is somewhat a realistic representation of a Southern Ocean diatom. But we wanted to push this a bit further and say, well, can we make these sorts of inferences by just seeing the proteome, but in an environmental context? So can we go into the Southern Ocean, specifically in McMurdo Sound, right in the edge of the sea ice, and take these metaproteome-derived proteomes and try and infer a set of growth rates. And here I'm showing you 2 weeks of metaproteomic data. So this is one sampling point at 1 week and one sampling point on the 3rd week. And this is another really, I think, powerful argument for biogeoscapes, because we were only able to do this on these 2 weeks because we had the associated dissolved iron and dissolved manganese concentrations that we could then run with our model. And so here, this is the inferred diatom growth rate from week 1 and week 3. And you can see that as iron goes from around 1 nanomolar to about half of that, the growth rate goes down. And that's not really surprising, but it is exciting that we're able to try and really get quantitative estimates for in situ taxon specific growth rates. But we can even try and push this further. Maybe we can infer the iron quotas per cell as the environment is changing as well. And again, this is taxon specific. So we're saying on average, diatoms have this iron quota, and we can look at other things as well, like the inferred iron uptake rate. And if you're interested in more of the metaproteomic approaches here, then you can, and looking at weeks 2 and 4, then you can check out this paper that we just published in ISME. So I'm really excited about this connection between metaproteomics and modeling to try and infer rates, but I don't think we should stop there and get so excited by this light, and really try and think what can we do with an ocean of rates and quotas, and what sorts of questions would we begin to answer with these new data types. For example, maybe these rates can explain differences in phytoplankton bloom progression across taxa, or maybe we can think of new ways to constrain biogeochemical models to improve their predictions. And with that, I'd like to end and just thank my supervisor, Aaron, and this work was done with a bunch of really great collaborators and funding. Thank you.
Thank you so much, Scott. That was a really great way to end the talks for the day. So thank you to all our speakers. I think what we're going to do now is we're going to try and take the few questions that we have in the chat for the speakers that just spoke and answer those live. And then we'll do another quick round of some Mentimeter questions that we can all digest the answers to together. And then if we have any remaining time, we'll go back to some of the lingering questions from our session earlier today. So maybe, let's see where we could start. We can start with a question from Clara Fuchsman. I don't know if Clara's still on the line. Clara, would you like to ask your question?
Sure, I can ask my question. Basically, we've been thinking about adding internal standards to metagenomics for a while, but we're terrified to do it because when you leave the euphotic zone, suddenly your DNA concentrations can get really low, and you're adding these internal standards before your extraction, which is because you need to do that for the information. So how do you know how much to add in deep water so that you don't accidentally just get standards as your metagenome?
So one thing you can do is, if these are precious samples, is you collect two samples. So you got your deep water sample, you collect 2 filters, and you extract your DNA off that one filter, and you get an idea of the mass that you're going to get. And then you can just tune down the mass of the internal standards that you're gonna get added to them. And so, what I can say is, starting when we did this in Marianne's lab almost 15 years ago up till now, we've never overwhelmed a sample. And part of that is, we added at less than 1% of the total mass to start with. but also the sequencing, as we've got more and more sequencing, you can kind of keep tearing down the amount of internal stand that you're adding and still get a good, robust signal. So so I understand the concern, but maybe the thing is just to take an example filter, do an extraction, see what kind of yield you're getting off that filter, and then tune how much you're adding to that.
Great, thanks, Scott. Ben Twining had a question for Julie Granger. Ben, would you like to ask your question?
Sure, yeah. Julie, loved the talk. Thank you. My question, I guess it's really about thinking as a program or a hopeful program, how far we go down the path of sort of individual group measurements, so to speak. You know, and I think you focused on some groups that are easily identifiable with flow cytometry. And you mentioned, you know, diatoms and coccolithophores that we also often pull out as having unique biogeochemical roles. But the minute we start looking at these omics data sets, we have all this other information about different groups that are present. And I'm wondering if where you sit in thinking about how far we need to go down those other paths of other types of, say, eukaryotic phytoplankton and what they're doing. And are there evidence from other studies that suggest we do need to go down those paths? Does that make sense?
Yeah, no, I think I'd like to go down the path.
Yeah, and see.
You mentioned, we think diatoms and coccolithophores have certain biogeochemical roles, and then we just don't think about anyone else, really. Those of us who think about the biological pump in just a process. So yeah, I would be fascinated to see if there are rules, like diatoms always suck up nitrate if there's nitrate available, whereas dinoflagellates don't really bother ever, or they do, or I want to know what the rules of engagement are, and if there are evolutionary strategies between groups or not, or if it's just chaos. I think some people could say more eloquent things about that, but I'm curious, certainly.
Yeah, great. No, I think that's a good argument, right? How do you know even yet whether we should go down there if we haven't gone down there? So, thank you.
I don't see any additional questions in the chat right now. Unless someone would like to respond to what Julie just said. I see Josue, did you want to ask a question?
No. I was just changing my mouse here, my setup in the computer. Sorry.
Okay, that's all right.
I just make one, just one response to that a little bit. Yeah, it was just yesterday, I think, that I saw an article, maybe just a forthcoming article in PNAS, which serves as some kind of cautionary tale. And they discovered a large proportion of trichodesmium cells are actually not diazotrophic. So it's not always as simple as linking functional groups to actual functions. So just something that we should be aware of. The heterogeneity within populations could be important.
Thanks for that comment. I think before we lose too many people, we're trying to launch the Mentimeter polls for the day. And then after that, we'll please feel free to stay engaged with the Zoom, and we'll go back to some of the questions from the morning session that we didn't, and the intercalibration talks that we didn't have a chance to ask. Heather? I'll also mention while Heather's getting set up there that another reminder that GatherTown is open and it will be open in its high capacity mode until at least 11 a.m. Eastern time tomorrow. And we're going to try and explore extending that. Okay, the Mentimeter poll is up in the chat now, so please feel free to take those questions.
Give us another 10 seconds. Intercalibration sitting right in the center there, along with funding and scope. All right. And one more question for today. What is the most pressing intercalibration and or intercomparison need? There you have it. It looks
Thank you,
like
Heather.
biogeochemical rates and metabolomics and organic geochemistry went out on this one.
Okay.
All right.
So
Go ahead, Adrian.
I was just going to say, so we should, so we have about 15 minutes left and for the time today. And so we do have some questions lingering from this morning's intercalibration update talks. So we'll get to those, but just a reminder that there is one more day in the workshop. Tomorrow we will meet here again at 1 p.m. And we do have a series of additional talks for you, as well as some more breakout activities. So please do join us if you can. So with that, there was a question earlier by Bethany Jenkins. Bethany, if you're still here, I believe it was for Mac.
Yeah, I was wondering, Mac, in the I had the I'd never sailed with McLean pumps until I was on this recent exports cruise, and I was new appreciation for how long it takes to process those filters. And so I was thinking about that in terms of transcriptomics and cells sitting on the filters. And I was wondering, in your calibration with the McLean collected samples, you compared sort of any timing between samples that came off the pumps or the first filters in the freezer or something like that. But just if you could share some thoughts on that.
Yeah, thanks. That's a great question. Let's see. I think the processing time, by the time we got the McLeans into the lab and open and started slicing, I think getting through the 4 filters was relatively quick compared to just the time they were down. You know, they pump for 3 hours or something. But I think it's a really good question for, you know, in general, like, is the processing time matter? For proteins, it's a little more forgiving, I think, than transcripts because they're so ephemeral. And do we want to design sampling systems that can preserve underwater? I think that's probably a better question. You know, like the AUV Clio does have the capability to preserve with RNA later underwater, which we know works for transcripts and proteins. We actually haven't tested that capability yet because we've been trying to do a lot of other engineering testing, but systems like that, I know there are other autonomous filtration systems that have been exploring that as well. So if that's a concern, that may help. Yeah.
Yeah, I was thinking about it, you know, the challenge of doing McLean pumps for like particulate trace metals at the same time as omics and the timing of that sampling. So I think for this, it might have to be one or the other off of a pump run unless we have some clever way of testing that.
Usually slice all the filters when they come up onto deck with, and get them into the freezers pretty quick. So I think it would be a shame to sacrifice, analytical opportunities, especially when the McLeans are so good at getting so much biomass or whatever underwater filtration system is used.
Yeah. Thanks, Mac.
Sure.
Can I just put a follow-up comment there? Mac and Bethany, also it's always striking to me, having been on these cruises, how long the pumps often, pump for. On GeoTrace's cruises, I think it's often like 8 hours or something, and or at least 4. So I guess also we might, as sort of an approach to the program, think about what is sort of the minimum or the max, minimum process that you might even approach, just based on how much the in situ process is changing over the sampling time before you even get it on deck. And it may be that's fine. We can say, well, we can't look at processes below 4 hours or half a day or something. Yeah, something else to think about.
Yeah, I think it's a great point. I mean, or some organisms like the diazotrophs in particular that we know have really rapid, transcriptome and proteome cycling. That is really difficult to do with that kind of system. For a lot of the algotroph bugs, microbes, they don't have as much sort of rapid regulatory response systems, and a lot of that stuff is tuned to sort of nutrients. So, maybe we've been lucky so far in that way of capturing, or maybe we're just measuring, we're distorting all the signals and we don't even know yet. But my personal belief is that I think we've been lucky so far to tease out important signals. But I think, yeah, it definitely should be systematically tested. Interested in other people's perspectives on that. I mean, I think it's a big challenge. Getting the samples right is an important effort.
Thanks, Mac. I don't want to put her on the spot, but Gabrielle Rocap had a question for Paul. Gabrielle, do you want to ask that question if you're available to, or?
Yes, I'm still here. And I have one more thought about Mac's comment first, which is it may matter more from different environments, depending on how different they are from that time aboard ship. We were talking about the deep sea versus the euphotic zone, and that temperature shift might be really different. Or, if the oxygen is changing, for example, that might matter more for some samples versus others. And I was lucky enough to be in the breakout group with Paul, so I got my question answered, but I'm sure other people would like his perspective on it, which is thinking about DNA extraction calibration and how do you decide what's best? And is it just the method that gives you the most DNA, which is what we're looking for, maybe for these low biomass samples Clara was talking about, but that's really good for short read stuff. And what about the DNA extraction protocol that gives you the longest DNA? Do we need more than one protocol? Even for that single DNA application, there may not be one size fits all. So I know Paul has thought about this. So
yeah, I think it's a really good question, and sorry, it's difficult to put all of this stuff into just a few slides, but those are things we have discussed, and I think it's important to have a sort of grassroots momentum, identifying other things that need to be tested. And that's really what it comes down to, is that we need to systematically test how these different methods might influence the downstream results. Whether or not you can have a single extraction method that's applicable to both long read and short read sequencing, or if you need to do several ones. But yeah, we need to test these. Short answer.
Go ahead, Scott.
Well, I was just going to build on, a thing that we've gotten with transcriptomics is this is related to Mac's comment, is that, over a decade plus of looking at transcriptome, metatranscriptomics now, we've never seen, or very rarely seen, something weird. And I think it comes to like, what is the true signal that we're going to look at? And so are we expecting that if we have something giving us a non-true signal, it's just going to be these huge stress response genes? So do we really know what, on a transcriptomics or proteomics side, what a off sample is going to look like? I think that question comes up a lot, is, how do we know what the true nature of these things are? In a way, almost related to what Gabrielle was just saying about like what is it, the true DNA community look like. But I think it's even more of a problem for transcriptomics and proteomics.
Thanks, Scott. Ben, you had one lingering question. I don't know if you still want to ask it about variability.
Yeah, I fear I'm definitely talking too much, but I'll throw it out there since I started. Yeah, I was just interested in whether the NAomics workshop group had thought about what level of sort of intercomparability is necessary, or what is a necessary level of comparability to achieve in order to answer the questions that we writ large want to answer. And you guys probably didn't, but it'd be great if you did. But either way, I think it's a good thing for us to think about, because we know there's going to be variability, but it may be that. Anyway, I'll stop talking. Is that something you guys talked about in your workshop, Scott or Paul or the rest of you?
I hesitate to throw this to Harriet, but she had that awesome slide about the
Oh, right, yeah.
splicing out, how many organisms you have in the sample, and then the sequencing is only going to capture part of that information, and then we could subset that further. But I mean, that's something that we have to wrangle with is what's the acceptable level of variability to answer the questions that we need. I don't know if anyone else on the workshop organizers want to comment on that at all.
And again, I think that comes down to finding the true sample. So if you're looking at a spread and variability, you got to have the mean or the point where you're anchoring that to. And I think we're at a point where we have a good way of even doing that. So kind of saying, well, how do you measure that variability? I mean, if you look at like Scott Jackson or other people that came to the workshop, when they were doing the human microbiome, and they said variability, that I think other people chime in, that meant you sent it out to all these groups and they got different answers on what a community composition looks like. And so I guess that's one measure of variability, is just how different on a kind of a similarity matrix will you get samples. And again, I don't think we have a good answer of what that should be.
Yeah, I know an interesting exercise we went through for NASA exports is for every measurement that we were taking, we had to provide a level of uncertainty. And so for a lot of these measurements, there has been some work to kind of figure out, oh yeah, if you do primary productivity, generally get, within 30% or so variability. I don't know if we really understand that much for the omics, in terms of the gene expression analysis, or even 18S or 16S or whatever, what consider to be an acceptable level of uncertainty for these measurements. So hopefully that could be something that comes out of these intercalibration activities.
Yeah, just understanding how different it is when the same group analyzes it 3 times. I mean, I don't know how often that's done, if ever, in sequencing, but it's interesting to think about.
I mean, so far we've been so hamstrung by the cost, and it's still expensive, but the cost is going down, and you can start actually thinking about, okay, we can actually do those kinds of experiments to see what we're dealing with. But yeah.
Well, thank you everyone for your really active participation today. I just want to remind everyone 2 things: that we would really appreciate if someone from your group could provide a summary sticky on your Jamboard of if you could distill your discussion down to 1 or 2 summary points on a sticky. A reminder that GatherTown is open. If you want to set a date with someone to go check out a poster this evening, you can do that. And we will see everyone tomorrow at 10 o'clock my time, 1 o'clock Eastern.
Thanks.
Thank you.
Thanks, everybody.
Thank you, everyone.